Welcome to the lecture on cell communication. In this lecture, we're going to discuss how and why cells talk to each other. How is it that cells can talk to each other or with other types of cells when they are both deaf and blind? If you think about a deaf and blind human, you know that we have ways of communicating with them. Oftentimes, these ways involve touch or feeling things so that they can actually understand with just touch what's going on around them. In this picture, you can see a protein that's made for touching. So these are like the outstretched hands on the cell membrane, and they allow the cell to grab on to things around them. If they find something that they understand the shape of, they can communicate that shape to the brain of the cell or the nucleus of the cell deep inside. Even single cellular organisms need to communicate with each other. What is it that they might talk about? In this particular picture, we're looking at two different yeast cells. A is one sex and alpha is the other sex. They're going to discuss whether or not they're mate compatible. In order to do that, they send out signaling molecules, much like a human would send out pheromones or use perfume. And the other yeast cell is then able to read that molecule, like we showed with the protein before. So if it fits inside of that protein, it knows that it has found a compatible mate. At that point, the two cells start moving towards each other until they can fuse into one cell. Myxobacteria are soil bacteria, and when they find that nutrients are low, they pull together their resources, they cluster together, and they form what we call a fruiting body, which has hard walls and is able to protect them for long periods of time. They do this through cell signaling. In multicellular organisms, cells need to communicate so they can work together for the full organism to be functional. In this diagram, you can see some local signaling that would happen between two adjacent cells. These cells can do cell-to-cell recognition, you can see in the bottom diagram, or sometimes cells that have to work really closely together will actually share fluids, so they share signal molecules directly between the cytoplasm. In the right-hand side, you can see that plant cells have plasmodesmata so that they can talk to each other across the cell wall. Local cell signaling doesn't have to be cell-to-cell. Sometimes cells can talk to other cells that are close by releasing either a local regulator, which goes out into the interstitial fluids or those clear fluids inside of your body and can talk to other cells close by, or by synaptic signaling, which is specialized for nerve cells in your brain, where a neurotransmitter like acetylcholine is released from one neuron, travels through the synapse, and then it attaches to the next neuron, where the next neuron is able to react and respond to the signal from the first neuron. But what if your body has to talk to all of the different parts of your body at once and really quickly? Your brain will talk to a gland, which will then oftentimes release a hormone, which will travel your entire body. Let's think about what a hormone might look like based on its structure related to its function. If it can't dissolve in the blood, it must be hydrophobic, and we know that all hydrophobic molecules are lipids. Right? So we're looking for a lipid. What's a communication molecule we've heard of that's a lipid? A steroid, right? So oftentimes steroids are released out into the blood system. They're hydrophobic, so they don't dissolve in the blood, and they have the ability to cross the plasma membrane, which is also mostly hydrophobic. Because of that, they can enter lots of different cells and can be very effective long-distance communicators. We call this endocrine signaling, as opposed to paracrine signaling, which is only to close-by molecules. Cell signaling and cell communication happens in three major steps. First of all, there's the reception of the communication molecule. Then there has to be the transduction, or the movement of that message through the cell to the target part of the cell that's going to respond. The third part is response. How is the cell going to respond? Is it going to make proteins? Is it going to move? What's it going to do? Now that we understand why cells communicate and understand the basic overview of how they communicate, let's get in a little bit more detail. This diagram illustrates a receptor protein. In this case, the receptor protein is a transmembrane protein, meaning that it goes all the way across the membrane. It has a signal molecule binding site, which will bind to a communication molecule called a ligand. Now, a ligand is any communication molecule. It could be a protein, it could be an ion, it could be any type of carbohydrate, it could be anything, but it has to fit in the binding site correctly or the protein won't recognize it. The protein then changes its conformation or changes its shape, and that change in shape is a signal to the inside of the cell. In this case, this protein recognizes a signal and is part of a G protein coupled receptor. When the G coupled protein receptor receives a ligand when that G coupled protein receptor receives its ligand and it changes its conformation, it allows the G protein, which is attached to its bottom, to be energized by GTP. GTP is just like ATP, except for instead of adenine triphosphate, it's guanine triphosphate. So don't be confused. So once that G protein is energized, it then slides along the interior part of the inner membrane, and it can activate an inactive enzyme. It activates the inactive enzyme by phosphorylating it. Once that active enzyme is phosphorylated, it starts the transduction process, or it starts to create a process where the message is then transferred throughout the cytoplasm of the cell. The word kinase is going to become important in this lecture. So a kinase is a protein that phosphorylates another protein. What does that mean? It just adds a phosphate to it, and whenever you add a phosphate to it, generally you activate it. So phosphates in your side of your cells oftentimes are on switches. In this case, this is a tyrosine kinase, which means it has tyrosine and it's going to phosphorylate stuff. When the signaling molecule binds to these receptor proteins, they actually dimerize. And we know di usually refers to two. So two separate proteins slide together. When they're super uber stable, slid together and holding each other upright, they can have the phosphates added to them. And those phosphates then act as on switches for a host of other proteins. The third major receptor protein that I want you to be aware of is a gated ion channel. So you can see that the ligand binds to the reception site on the gated ion channel, and the gate opens. Ions are then able to move very, very quickly from outside of the cell into the cell. And then once the ligand isn't there, the gate closes again. This will become really important when we start looking at muscles and muscle movements. Not all receptor proteins are in the cell membrane. Some receptor proteins are actually inside of the cytoplasm. When hormones are able to wiggle-waggle their way through the cell membrane because they're nonpolar, then they oftentimes are bound to receptor protein, which then escorts them into the nucleus and act as a transcription factor to initiate the transcription of genes. That sounds really complicated. All I'm saying is that not all receptor proteins are in the cell membrane. Some of them are in the cytoplasm, and they can help hormones get into the nucleus, where they can help the nucleus then do their job and respond to the communication. Transduction is the cascade of molecular interactions that relays a message from outside of the cell through the cytoplasm to the target molecules within. In this diagram, we see at the cell membrane, the orange stripe right there, we see the signaling molecule is bound to the receptor protein. This molecule this protein then changes in conformation and somehow activates a relay molecule. Now that relay molecule might be that inactive protein with the G-coupled protein receptor that was activated, or it could be one of those proteins that was activated by the tyrosine kinase. It could be a multitude of things, but whatever it is, it's going to start a cascade. That's kind of like how a rumor spreads, where you start off with just one really hyped up person, and then they tell 5 people. And then each of those 5 people tell 5 people. In this case, they're not telling each other exactly, but they're passing on phosphates. Now remember, when I said that phosphates act as on switches, so that's like turning on their friends, where their friends can go do some stuff and spread some rumors. Okay, so we call this a phosphorylation cascade, where once one molecule is phosphorylated, then it goes and turns on 5 others, and each of those 5 others can turn on 5 others. This amplifies the signal so that by the end of the pathway, the signal can sometimes be amplified as much as 10,000 times. When the signal is over or no longer necessary, that's when the phosphatases jump in and they pull the phosphates off those proteins and turn them back into their off position. So, kinases activate proteins, and phosphatases act at the end of the phosphorylation cascade to turn them back off. Not all transduction molecules inside of the cells are proteins. Sometimes your cells use what we call secondary messengers. Secondary messengers can be a second step, a third step, or a fourth step, so don't get confused. Secondary messenger just means pretty much I'm a messenger molecule, but I'm not a protein, which means that they don't necessarily phosphorylate anything, but they still can activate or deactivate molecules within the cytoplasm. One good example is cyclic AMP. Cyclic AMP is actually made out of ATP, which is then turned into a cycle form. In this diagram, you can see the first messenger is the ligand. It binds to the G-coupled protein receptor, which then activates the G protein, which then slides across the membrane and activates adenyl cyclase. Adenyl cyclase then creates cyclic AMP, or CAMP, from ATP. That cyclic AMP is then released into the cytoplasm, and it activates protein kinase A. Protein kinase A then starts a whole phosphorylation cascade. Another common secondary messenger would be the calcium ion. Calcium ions are actually in really low concentration inside of the cytosol, and that's because your cells actively transport excess calcium ions either outside of the cell or into the lumen of the endoplasmic reticulum. This diagram shows one example of the interaction between a signaling molecule and secondary messengers. In this case, the signaling molecule binds to the G-coupled protein receptor, which activates the G protein, which then activates phospholipase C. Phospholipase C destabilizes the PIP molecule, which then turns into inositol phosphate. Inositol phosphate then can bind to the endoplasmic reticulum's gated ion channel. Now, as you remember, when the receptor on a gated ion channel meets the appropriate ligand, the gate opens up. In this case, the calcium ions pour out, and they activate all sorts of other processes inside of the cell. This allows one signaling molecule to very, very quickly turn on lots of different cellular processes for maximum response. So far, we've discussed reception and transduction, but we haven't discussed cellular response. Cellular response can be turning on motor proteins so that they start to move vesicles filled with an enzyme to the surface of the cell so it can be excreted, or it can be the nucleus responding by initiating the transcription and translation of a gene into an active protein. This diagram shows a typical reception, transduction, and nuclear response, where a gene is turned on so that a protein is being manufactured. So what happens if you get an adrenaline rush? In this case, epinephrine is released quickly into your blood. It binds to a G-coupled protein receptor, which then activates the inactive adenyl cyclase Remember that secondary messenger, cyclic AMP? That's produced, which then activates protein kinase A and goes through a phosphorylation cascade. By the end of this, not just a few glucose molecules are released from glycogen stored within the cell, but thousands of glucose molecules are released from glycogen within the cell, and this provides fuel for your cells to produce ATP so you can quickly move in fight or flight response to whatever it was that caused that adrenaline to start pumping. Another example of effective cell communication would be apoptosis. Apoptosis is planned cell death. Planned cell death can occur because of either damaged or worn cells, or it can be part of natural development. In this case, a macrophage or a white blood cell is undergoing apoptosis. On the right-hand side, that you can see part of that process is it starts to form these little protrusions. Those are called blebs. Isn't that a great word? Blebs. So it forms these little protrusions, and then it eventually, those protrusions form pockets that are recycled inside of your body. In C. elegans, this is how normal cell death occurs, or apoptosis occurs. In the top diagram, you can see there's no death signal. And in this case, the CED9 protein inhibits the CED4's activity. So it's going to inhibit the other protein's activity. And CED4 and CED3 are now inactive. They're being held inactive. But what happens when the C. elegans realizes that it needs to kill this part of its tissue for development? Well, the body releases a death signaling molecule which binds to the receptor protein on the outside of the cell. That then activates a series of proteins that inactivate the CED9 protein. The inactivated CED9 protein now can't stop the CED4 and the CED3 from going crazy. They go crazy and they start turning other kinases on and other proteins on, which start to form the blebs and eventually start that recycling process of the cellular tissue. Now, when we think of cell death, we think like, oh, that's so sad, the cell's dying. But you wouldn't have fingers without apoptosis. This right here shows you the tissue of a developing fetal hand, and you can see that there had to be cell death in between the webs of your fingers so that you could actually have fingers, 5 phalanges. So, in this lecture, we covered why cells might want to communicate, the 3 basic steps of cell communication: reception, transduction, response. We went over several different examples of receptor proteins. We went over a couple of different ways that transduction can occur through phosphorylation cascades and through secondary messengers. And we talked about nuclear response and intracellular response. So, next class, we're going to have a lecture on the cell cycle and mitosis. How do cells know when to divide? I bet you it has something to do with communication.